So we'll get started on today. So I hope you guys all went well on the BIOC test on the weekend. I heard that it was not too bad, so hopefully you guys did well. So we're moving on to something a bit more, I guess, like moving on from just like all theoretical stuff onto things that actually like how things work in our body. So we're looking at digestion and the absorption of carbs and proteins today, and seeing what proteins and what enzymes are involved in that process. So first of all, we'll talk about energy balance. So of course we eat food because our body is always using energy, and so we need to eat food as a source of energy. And so in a perfect world, the energy that you eat will equal the energy that you expend. But in today's world, with Fatty Lane just around the corner, so it's pretty hard to do that. And so if your energy that's coming in is greater than the energy that's going out, then the extra energy is going to be stored as fat. So just there's 2 units of energy that we use when we're talking about energy. So that's that's the calorie and the joule. Okay, so 1 calorie is equal to 4.18 joules, and that's the amount of energy it takes to raise 1 gram of water by 1 degree Celsius. So that's why it's a weird number, like why 1 calorie is equal to 4.18 joules. So you've got your macronutrients. So you've got 4 macronutrients. That's your carbohydrates, your proteins, your alcohols, and your fats. And so you need to remember the at-water factors, because it comes up in your exam. So they might, in the finals, they might be like, oh, this person's eaten like 20 grams of carbohydrates or whatever. How much energy has he consumed? So then you'd just, if he had, let's say, eaten 20 grams of carbohydrate, then it'd just be 20 times 17. And whatever that answer is would be the amount of kilojoules that he's consumed. Okay, so carbohydrates at 17 kilojoules per gram. Protein is the same, 17, so 17. Alcohol is a bit more, so that's 29 kilojoules per gram. And fats is 38 kilojoules per gram. So you can see that fats are almost twice as calorie dense as carbohydrates and proteins. Okay. So we have our basal metabolic rate, which is the amount of energy, which is the rate of energy consumption while you're resting. So that's going to be different for everyone. Someone that has a fast metabolism will have a high BMR, which means that they could do nothing and still be burning a lot of energy. And that's going to be affected by like your age. So usually when you're younger, you have a higher BMR. And if you're male, usually because you have more muscle mass, you're going to have a higher BMI as well, and just your genes. And so it's pretty, I'm pretty sure it's come up in the final exam before, just things like what things would increase or decrease your BMR. So like if you're training a lot, then that's going to raise your BMR. If you're pregnant, you've got another person inside of you, so they're going to be using energy as well. So you're going to increase your BMR. A lot of it's like pretty common sense. Like if you think about it, it's pretty obvious. So if you have a fever, you're producing lots of energy, lots of heat. So that's going to raise your BMR. If you have caffeine, that's obviously going to get you going. And something called hypothyroidism which is where you're producing too much thyroid hormones, and that's going to elevate your BMI as well. And on the flip side, you've got, if you have malnutrition, so if you're not eating enough, your body's going to try and save energy, so it's going to decrease your BMI. If you're sleeping a lot, then your BMI is going to decrease. And so beta blockers are a drug, so they block the effects of epinephrine or adrenaline. So remember that adrenaline gets your heart rate up, and if you have a beta blocker, that's going to stop that effect. And hypothyroidism will also decrease your BMI. Okay. So then, if you wanted to measure your BMI, you could use direct calorimetry, so these measure your heat production. Okay. And then you've got indirect calorimetry, which uses respirometers, so they're things that you breathe into, and they're going to measure the amount of oxygen that your body has used by comparing the level of oxygen in inspired and expired air. And you're able to calculate the energy that your body has used, since the amount of energy is proportional to the oxygen that's consumed. Okay, so if you have food, so your body's main source of energy is fats and carbohydrates, okay? And when your body uses up, when your body burns those energy sources, you produce heat, and you produce carbon dioxide, and you also produce ATP. Okay. So you can calculate something called the respiratory exchange ratio. So that's just so the respiratory exchange ratio is just the amount of carbon dioxide that you breathe out compared. And so when you're burning your food, you also need oxygen to do that. And so the respiratory exchange ratio is comparing the amount of carbon dioxide to the amount of oxygen that you're using. Okay, so if the respiratory exchange ratio is close to 0.7, then your body is using fats. So if you look at the diagram at the bottom of your worksheet there, you can see that for fatty acids, you're using 23 oxygen molecules, but you're only producing 16 carbon dioxide molecules. So if you did, so that'd be 16 over 23, and if you did 16 divided by 23 in your calculator, you'd get something like 0.7. Okay. Whereas if you look at carbohydrates, you're using, you're producing 6 carbon dioxide and you're using 6 oxygen, so that's just 1.0. Okay. So by analyzing the air that someone's breathing out, you can calculate whether their body is burning carbohydrates mainly or fats. Okay. So the normal body at rest is about 0.8. So if it's 0.8, is it using more fats or more carbohydrates? Yep, so it's 0.8 is close to 0.7. So when you're just resting normally, your body's getting most of its energy from fats. Okay. So we'll move on to the next page. So remember that carbohydrates are one of our 4 macronutrients. So carbohydrates are a macronutrient. So it includes sugars and complex carbohydrates like starch. And a sugar is a simple carbohydrate. So it's either a monosaccharide or a disaccharide. And the advantage of the reason that it's simple is that it's only 1 or 2 molecules joined together, so it can be absorbed quickly. So there's 3 main monosaccharides in our diet, and that's glucose, fructose, and galactose. So they're in the picture below. So each of those are monosaccharides. And so glucose is the main monosaccharide, and it's the major source of energy for cells. And fructose can also be a major source of energy for cells. So fructose is more present in like fruit and stuff, and galactose is a monosaccharide that's present in milk as part of lactose. And if you look at the diagram below, you can see the glycogen molecule, which is how your muscle cells store glucose. So inside your muscle cells, you'll have glycogen, so that when they need energy, they can just break down that glycogen molecule to produce glucose. And starch molecules are found in plants. So plants store starch, and it's quite similar to glycogen. So the branched molecules, I'll just start again. So for starch digestion, starch is a complex carbohydrate. And then starch is how plants store glucose, and they're joined together by glycosidic bonds. And the branched chains are called amylopectin, as you can see in that diagram. And the unbranched chains are called amylose. So in order for carbohydrates to be absorbed, they have to be broken down into monosaccharides. Okay. Before they can be transported across the intestinal epithelium. Okay, so that's important to remember. You can only absorb monosaccharides. You can't absorb disaccharides or trisaccharides. It always has to be broken down to one saccharide molecule. So it has to be broken down to glucose, fructose, or galactose in order to be absorbed across your intestinal epithelium. Otherwise, it's just going to, if it doesn't get broken down, it's just going to stay in your intestines and you're just going to poop it out. Okay? Or we'll see what happens to lactose later, which is a disaccharide, so we'll see what happens there. So if we have an enzyme that's breaking down amylose, what would that be called? Yep, so amylase is the enzyme that's breaking down amylose. So that's pretty easy to remember. So that's going to break the glycosidic bonds between your glucose molecules. And there's, you've got 2 types of amylase. You have salivary amylase, which is released in the mouth. And you've also got pancreatic amylase. So that's made by your pancreas, and that's going to be released in your duodenum, which is the first part of your small intestine. So if you have let's say this is our let's just so this is our amylose molecule. So usually it'd be a lot longer. So we'd have amylase. So amylase will break these bonds. Okay. So if you, let's say this amylase enzyme just broke the bond in the middle, then you would end up with so if you broke the bond in the middle, you'd turn it into two glucose molecules joined together. So this is a glucose, this is a glucose, this is a glucose, this is a glucose, and two glucose molecules joined together is called maltose. Okay. So can maltose be absorbed by your intestines? No. So maltose is a disaccharide, so these cannot be absorbed by your small intestine. Okay? So if we go to the next page, we'll talk about disaccharide digestion. So maltose has to be broken down further into glucose for absorption to occur. So which enzyme is going to break down maltose? Shout it out. Yep, maltase. So you'll have the enzyme maltase, which is going to break that the bond between the two glucose molecules. And you'll end up with two separate glucose molecules, and now these are able to be absorbed by a small intestine. Okay? So you can imagine that if you're eating like simple sugars, like chocolate bar, it has lots of disaccharides. So disaccharides are pretty easy to break down into glucose molecules, so they'll be absorbed really quickly, whereas if you have a long, big branch chain of amylose or amylopectin or starch, so that's things like pasta, potatoes, things like that, contain lots of starch. It's going to take a long time to break it down into individual glucose molecules, so that's why it takes longer to digest if you have complex carbohydrates. So that's performed by the enzyme maltase, and so these disaccharidases are going to present on the brush border of the intestinal epithelial cells. So that's the microvilli of the intestinal epithelial cells. So if you have a this is your, so this is your enterocyte, okay, and here's your microvilli. So does everyone know what an enterocyte is? Have they used that word? So an enterocyte is a intestinal epithelial cell. Okay. So entero means gut, and cyte means cell. So it's an intestinal epithelial cell. Okay. So I'm just going to use that word enterocyte, and that means intestinal epithelial cell. And so the microvilli will be pointing towards the lumen of the gut. Okay. And so the lumen of the gut is where all your sugars and things will be. So your enzymes, so your enzyme will be present on that, on the brush border of that enterocyte. And the reason that the enzyme is there is because once it breaks down those disaccharides into monosaccharides, if it's right next to the surface of the cell, then it's going to be absorbed really quickly. Okay, so that's just facilitating the absorption. Yeah, and the reason that it's called a brush border is because if you look at it down a microscope, it looks like the end of a brush. Okay, so sucrose is a disaccharide, and that's made up of one glucose and one fructose molecule. So sucrose is the sugar that's in your table sugar, like white sugar. And lactose is a disaccharide that's made up of one glucose and one galactose molecule. Okay, and so you have the chart there, which shows you which enzymes are breaking down which disaccharides, and that's pretty easy to remember. So sucrose is broken down by sucrase, lactose is broken down by lactase, maltose is broken down by maltase. And I think the most famous one of these would be lactase. So I think maybe some of you are lactose intolerant, or at least one of you will know someone that is lactose intolerant. So when you're a baby, everyone produces lactase, because if they didn't, they wouldn't be able to digest milk, and that'd be pretty bad. But as some individuals get older, they don't produce as much of this enzyme, the enzyme lactase, and it just depends on your genetics. So if there's not enough lactase to break down the lactose, then the lactose, which is a disaccharide, it's not going to be digested, and it's not going to be absorbed by the small intestine. And then, so if it's not absorbed in your small intestine, it's going to go all the way into your large intestine, and in your large intestine there's usually a lot of bacteria, whereas in your small intestine there's not a lot of bacteria. So if if that lactose gets into the large intestine where there's a lot of bacteria, the bacteria are going to break it down, and when the bacteria break it down through the process of fermentation, they're going to produce gases and acids, which causes the symptoms that you get from lactose intolerance, which is farting a lot, that's flatulence, having distension, so bloating, and abdominal pain. Okay, so that diagram on the left for the lactose tolerant person, that's not exactly quite right, because so here's your Oops. Because that diagram on the left makes it look as if you're absorbing the disaccharide, but remember that you don't absorb the disaccharide. So if you had so this is lactose and this is lactase. So the lactase enzyme is going to break down the lactose in the small intestinal lumen, and you'd have these monosaccharides in the small intestine, which would then be absorbed through the epithelium of the intestine as monosaccharides. Okay, so just that diagram on the left is a little bit wrong. So just make sure that you understand that it's only monosaccharides that are going to be absorbed. Okay, and you have that diagram on the bottom. So I just want to show you So I just want to remind you about, I'm going to talk about the apical membrane and the basal membrane and the lateral membrane. So here we have two enterocytes. So we have the microvilli. So the microvilli are present. So this here is the apical surface. So the apex is pointing towards the lumen. And this part here is the basal surface, and this part here is the lateral surface. Okay, and these enterocytes are going to be joined together by proteins. Okay, and these are tight junctions. And what do these tight junctions do? What's their purpose? No. So you've got things in the gut. Do you want to let anything through that epithelium? So you're supposed to have a barrier, because remember that the lumen of the gut is essentially the outside world. So you have to be very selective about what you're going to let through. These tight junctions are going to prevent anything from getting in across that epithelial membrane. Okay. So water and salts like sodium and potassium can get across that, those tight junctions because they're very small molecules. But normally big things like proteins aren't going to get across there. So we'll move on to the next page. So carbohydrate absorption. So I've got it again there. So only monosaccharides can be absorbed by the small intestine. I'm just repeating that because, yeah, it's just important that you remember that. So sugars are polar molecules. So if you remember, you have your glucose molecule, and you have like all these hydroxyl groups on them. You have a lot of oxygen, right? So that's going to make it polar. And so they can't diffuse across the non-polar cell membrane. So they require a transporter protein in the cell membrane to get absorbed. And that transporter is called the sodium-dependent glucose transporter 1, which is present on the apical membrane of the enterocyte. Okay, so I've just got a diagram there, and I just want to go through it. So you've got so here we have the intestinal lumen. Okay, so your food's going to go through here. And then you have so this is your enterocyte. Okay, and then on the basal surface of your enterocyte, you're going to have your blood vessels. Okay, so this is your blood vessel. All right. So you've just eaten a meal, and so if you've eaten a normal meal, it's going to contain carbohydrates, it's going to contain fats, it's going to contain proteins, but we're just going to focus on the carbohydrates for now. So let's say you'll have eaten some starch, and then the starch will get broken down into, let's say, maltose. And then the maltose would get broken down into glucose, right? So here's your glucose molecule. Now, glucose can't get into the enterocyte on its own. So we have a transporter on the apical surface of the enterocyte, and this protein here is called sodium-dependent glucose transporter type 1. So in order for glucose to get into the cell, we also have to have a sodium molecule. So we have to absorb one glucose for every sodium molecule that we absorb. Okay. And the reason for that is that normally, so like as soon as you start eating, you're going to start to absorb glucose into your enterocyte. So normally, the glucose concentration in your intestinal lumen is usually kept quite low, okay? Because you're going to absorb it as soon as it forms. And the concentration of sodium in your intestinal lumen is usually going to be relatively high, okay? So that's high in comparison to the enterocyte. So in your before a meal, the glucose concentration in the enterocyte is low, but then once you've started to absorb glucose, then quickly the glucose concentration in your enterocyte will become high, okay? Because remember that you're absorbing all this glucose into your enterocyte. And so then what's the problem here? If you've got a low concentration of glucose in your intestinal lumen, and you've got a high concentration of glucose in your enterocyte, then can glucose just simply diffuse into your enterocyte? So it won't, because it'll be going against its concentration gradient. So remember that molecules always want to diffuse from high to low concentration, but in this case it'd be going against the concentration gradient, going from low to high. So we need to have a mechanism to allow the absorption of glucose, even though we're going against the concentration gradient. So the way that our bodies develop to do that is to couple the absorption of glucose to sodium. So the intracellular, so inside this enterocyte, the intracellular concentration of sodium is going to be low. Okay. And so does anyone know how the intracellular concentration of the enterocyte is kept low? So we have an enzyme. We have an enzyme on the basolateral membrane of the enterocyte, and this enzyme is called sodium potassium ATPase, sodium potassium ATPase. So this maintains the low intracellular concentration of sodium. So it's going to take 3 sodium and pump it out into the bloodstream. And in exchange for the sodium, you're going to get 2 potassium and pump it inside the cell, because remember that the potassium concentration is high intracellularly. And to do this requires energy, because it's against the concentration gradient. So we need to have ATP. So ATP will be used up in this reaction, and ATP will be converted into ADP. Okay, and that's going to provide the energy. So yeah, if we if we look at the blood vessel, remember that the glucose concentration inside your blood is kept pretty low at about 5 millimoles per liter. So the glucose concentration in your blood vessels is relatively low. Unless you've got diabetes, then it'll be high, but let's just assume that you're healthy. And then the sodium concentration in your blood, so in your plasma, is usually high compared to the intracellular concentration. Okay? So then, once we've absorbed this glucose into the enterocyte, so remember that there's a high concentration of glucose inside the enterocyte. The concentration gradient is downhill from the enterocyte to the blood vessel. So you can just have a different type of transporter here, and this one is just called GLUT2 or glucose transporter type 2, and this one just allows the passive diffusion of glucose down its concentration gradient into the blood vessel. Okay? And then this blood will go through the capillaries of your skeletal muscle. And so let's say further down you have a muscle cell. Okay, so the muscle cell has a glucose transporter, and this one is glucose transporter type 4. And this one is insulin. So this one is insulin dependent. So these muscle cells will only have this transporter on them if you have insulin. Okay. All right. So we'll just go through the bullet points at the bottom now. So after a meal, the concentration of glucose inside the enterocyte rises quickly. This would mean that absorption of glucose into the enterocyte would be difficult since it has to go against the concentration gradient. In order to allow glucose absorption to continue, the sodium-potassium ATPase on the basolateral membrane, so on this membrane here, it's going to pump sodium out of the cell in exchange for potassium, and this uses energy in the form of ATP, and this maintains a low intercellular concentration of sodium. There's a downhill gradient for sodium into the cell from the intestinal lumen, and the sodium-dependent glucose transporter type 1 protein couples the transport of sodium down its concentration gradient with the transport of glucose against its concentration gradient. So that's this one here. And then glucose transporter type 2 on the basolateral membrane, so that's this protein here, allows the diffusion of glucose out of the enterocyte into the bloodstream. And then we have our other tissues, such as our muscle cell, which will have, so the muscle cell will have an even lower concentration of glucose than the blood. So that means that if you have this GLUT4 protein on the muscle cell, glucose will diffuse from the blood into the muscle cell, and then once the glucose gets into the muscle cell, what's going to happen to it? So here's our muscle cell. You have glucose, which gets into the cell, and then so we have glucose inside the cell, and then the muscle cell is going to convert that into glycogen, right? Because it's just going to store, unless the muscle cell is very active at that moment, then it'll just use it up. It might break it down into ATP, but otherwise if it's just resting, it's going to it's going to turn that glucose into glycogen. So that means that the intracellular concentration of glucose inside the myocyte, or the muscle cell, is always going to be kept very low, lower than the blood, right? Because anything that comes in is either going to get broken down into ATP or turned into glycogen. So we're going to go over this stuff a lot more later on in the course, okay? All right, we'll go on to the next page. So then we'll talk about protein digestion. So proteins in the food are broken down into individual amino acids, dipeptides or tripeptides. So the intestine doesn't allow peptides longer than 4 amino acids long to be absorbed. So what would the problem be if your intestine was just absorbing big proteins whole? So if you were, let's say if you, I don't know, ate a pancreas or whatever, and it contained a lot of insulin, then if you were absorbing the protein whole, then it could get into your bloodstream and then start doing things, because most of these proteins are going to have a function, right? And they would start doing things that you don't want it to do, right? So you want to break it all down into peptides that are short, so that means it loses all its function. Okay. And so proteases and peptidases are the enzymes that break down the proteins by breaking down the peptide bond, and that's via hydrolysis. And these are going to be zymogens. So zymogens are enzymes that are released as inactive forms and then cleaved to become active. So most of the proteases that you release are zymogens, and that prevents you from digesting yourself. Okay, so you only want it active in the intestinal lumen, not inside the pancreas, for example. So pepsinogen is released in the stomach. It is activated by low pH, so the acidic environment, which causes the exposure of the active site, and then the active site cleaves itself to activate it. So the active form is known as pepsin, and so this is an example of a zymogen because it's released as an inactive form. So your pancreas is an organ that's situated right next to your stomach and small intestine, and it produces enzymes. So which enzyme does it produce that breaks down carbohydrates? Yep, so pancreatic amylase. So remember that the pancreas also produces enzymes that can break down carbohydrates, and also it'll produce lipase as well, which is an enzyme that breaks down fats. But right now we'll just focus on the proteases. So there's a few different types of proteases. You have trypsinogen, chymotrypsinogen, and procarboxypeptidase, and there's more. Okay, so you have basically 2 types of proteases and peptidases. So you have your polypeptide. So let's say this is just a polypeptide, a peptide chain that's 6 amino acids long. Okay, so if you have a endopeptidase, it's going to attack the peptide bonds that are in the middle. And if you have an exopeptidase, it's going to attack the bonds that are at the end of the peptide. Okay, so examples of endopeptidases are chymotrypsin, trypsin, and pepsin. So these are all endopeptidases, so they'll attack the peptide bonds in the middle of the peptide, whereas exopeptidases, you have carboxy peptidase, and you have amino peptidase. Okay, so if you remember, this side's the N-terminus, which has the amino group, right? And this side's the C-terminus, which is the carboxy group. So this one would be carboxy peptidase, and this one would be amino peptidase, right? Yeah. And then, so the absorption of amino acids is pretty similar to the absorption of glucose. So it's also dependent on sodium. So remember that. It's a little bit different if you have a dipeptide or a tripeptide. So remember that you can absorb peptides up to 4 amino acids long. So if it's 2 amino acids joined together, that's a dipeptide. And that's going to be absorbed with a proton. Remember that you're going to have a lot of protons in your intestine, remember, because your stomach's secreting a lot of protons to make it acidic. So there's going to be a lot of protons in your intestines, which can then be absorbed with your dipeptides and your tripeptides. So there's a special case where you can absorb peptides longer than 4 amino acids, and so that only occurs in newborn babies because their intestinal tight junctions aren't properly developed yet. So remember, the tight junctions were the proteins between the cells, which were acting as a barrier of things getting through. So this allows the absorption of maternal antibodies. So remember that antibodies are proteins, okay? And if you broke them down, then they would lose their function. So in the newborn baby, this allows the absorption of maternal antibodies, which can then be absorbed into the bloodstream and fight against infection. Okay, and then we'll just quickly finish off with DNA, RNA digestion. So remember that if you're eating a plant, the plants are made up of cells, and every cell has DNA and RNA in it, right? And if you're eating meat product, then that's also going to contain cells, and that will contain DNA and RNA. And so DNA and RNA, you don't actually get any energy from them, okay? But you can use the DNA and RNA, you can just break it down and use it as building blocks for your own DNA and RNA. Okay, so you have nucleases, which will hydrolyze the phosphodiester bonds, which connect the nucleotides. So if you look at that diagram below, you've got a phosphate molecule, so the phospho, connecting the 2 nucleotides with 2 ester bonds, so 2 esters, diester, so phosphodiester bond. Okay, and then we got celiac disease. So celiac disease is when the immune system is reacting to gluten and produces antibodies. These antibodies are then modified by an enzyme called transglutaminase. I wouldn't bother remembering that enzyme. And then these modified antibodies will attack the cells in your small intestine. So the enterocytes are going to be attacked by those antibodies. And if they get attacked, they're going to die off and lose their microvilli, and the structure will also be changed as well. So then remember that you need those microvilli and villi in order to have that surface area for absorption, and you'll get malnutrition. So celiacs need to avoid gluten. And then we also have cystic fibrosis. So one of the complications of cystic fibrosis is that you have very thick and sticky, or in other words, viscous pancreatic secretions. So remember that all your pancreatic enzymes has to travel through your pancreatic duct in order to be released into the duodenum. So if it's very thick and sticky, it's just going to block up that duct and it's not going to be able to get through. So that means that there's no enzymes in your intestine, so no amylase. So if there's no amylase, your amylose isn't going to be broken down into glucose or monosaccharides. So then remember that if you're not breaking carbohydrates down into monosaccharides, you're not going to be able to absorb any carbohydrates. If you don't have chymotrypsin, trypsin or chymotrypsin or trypsin, then you won't break down your proteins. If you can't break down your proteins, you can't digest, or you can't absorb those proteins. And you also won't produce lipase, which is going to break down your fats. Okay, so that'll cause malabsorption. Okay. And so you can treat this by taking supplements that contain those pancreatic enzymes. Okay. Yeah, so that's it for tonight. So I think we're covering like fat digestion and other stuff next week. So see you then. Cheers. Thank you.